this is just Golgi apparatus that's been sectioned, so it's been cut in half. So we know that the Golgi apparatus is the final port essentially for those vesicles of proteins that are going to be moving out of the cell. These are vesicles, okay? And these are also vesicles, vacuoles. Vacuoles are simply large vesicles. So then we come over here on this side, and this is showing us those cell junctions that we talked about. These are tight junctions here, so these basically form a seal. And then these are supposed to be gap junctions. And then down here, which is not really great, these are supposed to be desmosomes. So those would be those cell junctions that are like Velcro, okay? These are microvilli, so this would be a small intestine cell, and those are cell extensions that result in the absorption of nutrients if it's in your small intestine. So that's an animal cell. So then we look at So we're looking at this, and so this is, once again, we've got a lot of similar structures, nucleolus, the nuclear, this is called nucleosol, and so that's got chromatin fibers in it, a nucleus with nuclear pores. This is a plant cell. So then here we have our rough endoplasmic reticulum, Golgi apparatus, but the unique thing is that we have a chloroplast, and then we have vesicles, and then we also have something that animal cells don't have, and that is a large central vacuole that holds water. If we look at the sides of a plant cell, there's a plasma membrane, but there's also a cell wall, right, that's composed of cellulose, and then we have these little pores, okay, and those little pores are called desmata, okay. So that's for that. Now, if we come over here, these are cards that go through the process of mitosis, okay? And so what everyone should do is play with these cards and be able to organize them in the appropriate fashion, because that would probably be part of your practicum. And then we come over here. Now we're looking at mitosis and these models. And so let's say I would number these, and you would have to put them in the correct order. So you'd want to know these models well. So this is going to be interphase. And so I'm looking at this, and look, here I have my centrioles, and they're just in one place. And I only have two little centrioles in that centrosome. Here's my nucleus, here's my chromatin fibers. They haven't coiled yet. And then I have my other cell organelles there. And my nuclear membrane is intact. So then I move into prophase, and then in prophase, here I have my centrioles, but I don't just have a single pair now, I have two pair, right? So we've gone through interphase, and we've made these organelles, so we have duplicates of everything. So now I look at my chromosomes. My chromosomes are starting to look thicker, and my centrosomes I have doubled. My nuclear membrane is still intact, that's prophase. And then this would be prometaphase, okay, prometaphase. So my chromosomes are thickening, my nuclear membrane still intact. I have my centrioles are moving towards either side of the cell. And we've got duplicates of all the organelles there. So now I am at metaphase. My chromosomes are in the middle. Oh, and I should say too, this represents the spindle that is formed, and so the spindle is starting to form between the centrioles. Remember, the centrioles make the spindle fibers that are going to organize my chromosomes. So now here, my spindle is formed. These are all microtubules. They're all attached to those